what I want to talk to you about really is an overview of genomics, explain what it is a bit, and then also what it is at Cold Spring Harbor, because as Bruce said, it's a bit different. And it's really genetics, although it's genetics in a way that we now view the world where we can look at all of the genetics that's happening in an organism. So I want to start off with really some basics of what is genomics as opposed to genetics. It's really looking at the structure and function of all the hereditary material, and I'll talk about this on the next slide. Basically, the structure and function of the genomes, and I'll explain again what that is in the next slide. The main tool for doing that is something called DNA sequencing right now, and that is a series of chemical reactions that now are run through highly automated machines that let us determine the order of the 4 bases that comprise all of our hereditary material, the A, C, T, and G, adenine, cytosine, thymidine, and guanine. And this technology has expanded so much, it's really become a centerpiece in a lot of molecular biology work. And much of what I'll actually talk about is how that happens. And not how it happens in the lab, the biology, the chemistry of it, but rather how it impacts the biology. The human genome is, as I said, our hereditary instructions. And it's about, for each of us, about 3 billion of these A, C, T's, and G's in a particular order that encodes the instructions for making us. It's our biological blueprint, if you will. And so out of this 3 billion bases, there's about 1 or 2% that are actually a code that signals our bodies to make the 20,000 proteins that exist in us. And it's by the order of these 4 bases that is encoded. So if you think of it as just a book, except only 4 letters, it makes this iterative code as it instructs the body to make the proteins in the 20,000 genes. And so these bases, the order of them determines what amino acid gets put in the protein. So it's like a code in the hereditary material that really makes us. A version of the human genome, what's called a human genome reference, was published in 2001 by two groups, actually. One a commercial effort, and the other a government-funded, foundation-funded effort to publish a version of the human genome as a reference. And it's important to realize, as you'll see on the next slides, that this is only a reference. This actually doesn't represent, there's no human being with this sequence. It's actually about 70% one anonymous person from Rochester, New York, and the rest range from a few other people to the occasional postdoctoral fellow who drew blood and made a library and sequenced a few clones. So it's mostly this one anonymous person from Rochester and then a bit of everything else mixed in. I wanted to give you some idea of the scope of 3 billion bases. So each of our genomes encode our hereditary material. And one way of looking at it is that basically, it took me a whole afternoon to figure this out on Google and counting words and stuff. And so I actually did it initially. I was making a presentation by Skype at the Science Club in Orlando for DNA Day, and I figured everyone that was at the Science Club had probably been to Universal World before they went there. So I decided to use this analogy. But basically, if you think of it in terms of the Harry Potter books, one human genome is a stack of those books about half as high as as the Empire State Building. So that's one human genome. And so what the machines we do now that I'll talk about do is determine the order of the letters in that very tall stack of books. Our genome, as I said, is about 3 billion bases. To put that in perspective, these are the genome sizes of a number of other genomes. This is a relatively large virus, about 300,000 bases. Bacteria are in the 5 million range. The smallest complex eukaryotic organisms are about 15 million. The yeast, this is what makes bread or beer or soy sauce, what have you. And then they start going up. Some of our favorite model organisms are about 150 million bases. And then you get up into the size of mouse and human, which are about the same. Chimpanzee is almost exactly the same as human. Interestingly enough, most plant genomes are actually very big. And as an example, the wheat genome is about 5 times the size of the human genome. The loblolly pine genome, this has actually gone up a little. It's about 22 billion bases. I'm on their advisory committee. There's a group funded by the USDA sequencing this, and it's drifted up. Loblolly pine, if you're writing on something or have a piece of paper, that was probably at one point a loblolly pine tree. That's why it's interesting. But it's a very large genome. As I said, the genome that came out in 2001 was a reference, because with each of us, our genome differs from that at about 1 base out of every 1,000 to 1,500. So basically, our differences, our biological differences in our hereditary are created by these differences. There's also some bigger differences that sometimes affect us, the type thing that Mike Wigler has looked at a lot. But there is this single base change about every 1,000 to 1,500 bases. To put that in perspective, we've recently identified a single one of these 3 billion bases that changes a single amino acid in a protein that probably makes people have an extremely high risk for bipolar or major recurrent depression. We've also, other people have found, for instance, that there's, out of that 3 billion bases, 66% of Caucasians with cystic fibrosis have the deletion of 3 of those 3 billion bases that causes them to have cystic fibrosis. So very, very small changes can lead to devastating consequences. Or they can lead to 2 animals which are essentially the same but look radically different because of the base changes. These are both dogs, obviously, and dogs are a little special because they have the highest degree of diversity for the same species among any mammal. But you can see these are really the same, and the differences between these are some number of changes in the nucleotides in their genome. People have been trying to do this, as Bruce said, this is really genetics, for a long time. And what we find here is, this is an example I wanted to use. This is what it was 20 years ago, it's right before I came here. This was an effort to try to find the gene that causes Huntington's disease. And so what we did is we sequenced 58,000 bases. 30,000 of that came from 2 different people. They overlap from different clones in the lab. And in that overlap, we identified 72 differences between the 2 individuals. And this was actually a big project. If you're familiar with some of the people, this is the current director of the National Institutes of Health. This was Craig Venter. This was probably a $3 to $4 million project to sequence 58,000 bases in 2 people in 1992. I'll come back to that. The first human genome was sequenced, as we said, in 2001. It was the culmination of about a 10-year project of concerted effort. And it was mostly done the last few years. The cost estimates are kind of all over the place, but $100 million to do that one genome is a relatively good estimate. And that's, again, an important thing to remember as we move ahead and see what's happened in recent years. So that one reference human genome was sequenced using what we call old technology, because it is now. And it required a lot of sample preparation. We had robots kicking out tens of thousands of samples a week to do that in our lab. Machines got loaded every day. One of the machines would generate about 1 million bases a day. And so places around the world had anywhere from dozens to hundreds of these machines, each kicking out 1 million bases a day. But around 2007, this drastically changed. And it ultimately changes the way we do biology in a lot of ways. The new sequencing, which I'll talk about in general terms, is what we call massively parallel. So rather than do a single molecule at a time, basically we put in a whole genome broken into fragments. And that cuts down the preparation time, and the machines run longer, so we don't have to load them as much. The initial version of this machine, as opposed to 1 million bases a day, would in about 2 weeks produce 27 million sequences of about 36 bases each, so about 1 billion bases. So basically went up 1,000-fold. in roughly at least 100-fold. These runs were shorter. These have continued to improve, and the current version, this has actually gone up a little. On a good run, we're pulling about 740 billion bases per run. So compared that to 1 million bases per run. And it's really been a completely transformative change in the technology that lets us do things that we really could not have even conceived of as recently as 2007. To look at that in some other ways, how long would it take a person, or how many people years would it take to sequence a genome? When sequencing first really started, sort of late 70s, early 80s, estimated about 3 million years to sequence one human genome. That continued to drop. Even before the advent of this thing, this new technology in 2007, it was about 50 person years to do a genome. And then in 2009, by the time these had started to ramp up, it had dropped to about 14 days. And we now do, currently in our lab, we're quite capable of doing, with one person, 6 genomes every 13 days on a single instrument, and we have 9 of those instruments. It takes more than one person to run them, but so again, it lets us, and I'll come back to this, it lets us look at science differently than we used to. Another way to look at it is cost. This is from the National Human Genome Institute. This is the cost of doing a genome, the dots. This is Moore's law, named after Gordon Moore, one of the founders of Intel, who came up with the postulate that you could basically pack twice as many transistors on an integrated circuit every roughly 18 months. Moore's law is often used as a metric for computational acceleration. And you can see up until about 2007, when this next gen started, cost was progressing about the same as Moore's law, which everyone thought was about as good as possible, and then it just fell off a cliff in 2007. This stopped in 2011 with a cost of around $12,000. Current cost now is roughly, it's a bit under $3,000. So it's continued to drop. And that's down from 100 million 10 or so years ago. This has had broad impacts at Cold Spring Harbor. One area, and I'll talk about several areas separately. One area is human and model organism genetics, both structural and functional. Structural, I mean looking at the structure of the genome. Functional, looking at the function of it. The stuff in blue is with old technology sequencing. The stuff in sort of pink is new technology. So we were involved. We were one of the 20 labs in the world that were involved in that Nature publication from 2001 on the sequence of the human genome. We also were involved in the mouse genome sequence and the fission yeast genome. It is probably an indication of how the technology moves that this one was started before the other two. Just because you get started and all of a sudden something new comes along and you just walk by it. Another example is, as Bruce mentioned, plant genomics. We've actually been very active in that at the lab, starting with donation of sequencers, actually for specifically plant genomics. And then we were involved in our lab in the sequence of the first plant chromosome from a plant model organism named Arabidopsis, then the sequence just a year later of the entire genome. And then with Rob Martins, and all these projects were actually with Rob, in these early ones at least. And then we're involved in new technology to analyze, again, these very, very large plant genomes. And then involved in the rice genome and the corn genome. More recently, the technology has changed dramatically, as I've been talking about, and we've been very much involved in trying to use that technology, which I've told you how great it is in terms of numbers of bases, but it has some technical limitations I don't want to go into the details of, but it makes it difficult to do new genomes as opposed to redo a genome. And so we've been trying to get around those problems with new strategies and new approaches, and have been applying those to cocoa, tomato, and more recently wheat, and those are ongoing efforts here at the lab. In addition to sort of genomics, and this sort of genomics genetics kind of merges, it's also been involved, having these technologies have been involved in human genetics at the lab. So the gray things here are things Mike Wigler has done actually with arrays or a different technology. Blue is old generation technology, and the pink is, again, new generation technology. And the reason I want to put these up here is to stress the fact that because we have this technology here, if somebody has an idea, and they're likely to have the idea because we talk about these technologies and what they can do, that we can really pull the trigger on it very quickly. Here's an example of something, the RNA libraries done by Greg Hannon. This was actually a case that it wasn't really what you typically think of a sequencing project. It was to make a bunch of these clones. But in fact, the way we actually figured out it was easier to make them was to make them randomly almost, and then sequence them to figure out who was who, because it was that much cheaper, even with the old technology, than making each one in a directed fashion and checking it. So it went from, instead of sequencing each of the DNA fragments that need to be sequenced for this in a tube, sequencing them on an array, which is vastly cheaper, cleaving them off, cloning them, and then sequencing to see who was who, because that was easier than putting each one in a tube to begin with. But only because of the technology was so good. When we started next generation sequencing, that really kicked things off again. I mentioned earlier that about 3% of the genome makes proteins. So Greg Hannon and I actually developed the method we call exome sequencing to capture, and it was actually based on some of the same core technology as used up here, but to capture that 3% and sequence that 3% from a person. So it's obviously 30 or 40 times faster than sequencing the whole thing. And that's been really taken off. We had really the first publication on that in 2007. People have enhanced that and do it different ways, but last time I checked, there have been over 1,000 papers using that technology of exome sequencing and usually studying human diseases. And then also having the technology, working with Mike Wigler, we worked on looking at tumor cells, single tumor cells. And then lastly, as an example, with Mike again, looking at mutations in children with autism whose parents don't have autism, and looking in their protein coding region for differences that might attribute that to autism. And I should add, we've also more recently done this with a smaller set of samples, but from schizophrenic patients, so patients with no parents that don't have the illness, and the child is schizophrenic. And interestingly, we're seeing a fairly broad overlap in the genes in autism and schizophrenia, which Bruce alluded to earlier. So this technology, which is really a core technology, has really impacted a lot of biology here at the lab. And one of the reasons for that is when it gets down to it, these machines basically count molecules a billion at a time. So anything you can sort of convert to an assay where there's a DNA fragment that results from it, or an RNA fragment, you can actually count them on these. And so this has really impacted a range of biology at the lab. When I first came here, there was probably about 5% of labs at Cold Spring Harbor that were doing anything with genetics or with genomics, and now it's about half. So it really has, over the years, impacted the lab broadly. Some examples of that, of how, again, having this technology let us move quickly in a pretty competitive environment. One is, again, I mentioned the exome sequencing, which has been used in a lot of illness, looking at disease genes, both here and at many other places. The single cell work, which gives, I think, a different perspective on what a tumor is, because it helps us elucidate the heterogeneity in the tumor. Not each cell in the tumor is the same, and those differences have potential impact for understanding cancer and also for therapy. The RNAi technology, as I mentioned, really requires the sequencing technology just to drive it to make it happen. And then, again, counting molecules in the work that Bruce talked about from Chris's lab, being able to count molecules kind of at will and very effectively was an important part of that project as well. If we look at the output of sequencing over the years, you can see this dramatic increase from 2007, and we're kind of plateaued here. This is the number of lanes we count. This is machine use, but it shows how it's really risen as we've had the technologies available. That's even more dramatic when you look at the output in sequencing, because not only are we running more lanes, but each lane over the years is producing more data. So this is, for someone that's been doing this when we were, throwing around radioactive stuff and trying to get 300 bases off one gel, to see this number, which is 90,000 billion bases, still is something that doesn't quite compute for me. Our best per month is roughly 10 trillion bases per month. And that is, again, just a game changer in how we do biology. This shows sort of where other places are. This is a little outdated. I should point out that the New York Genome Center has now actually, they have more machines than us. They've gone a bit beyond us. But we're really a leader in this part of the country. Actually, in the US, we're one of the larger centers in the US, probably the top 10. Because, again, of having this technology, when the stimulus came out and the extra funding available, we were well positioned and were able to secure about a bit under $11 million in research grants, because we could put in grants saying, we have machines here on site, we can do this now, and did very well in that competition, startlingly well, actually. And again, a range of biology being impacted by that, but core to the lab's mission in all cases. As examples, again, of this, and I want to compare, I put this up here to compare it to that earlier one I showed. So that was 20 years ago, 50,000 bases roughly in one person and about 30,000 for another person. This was a paper published last year where we looked at 500,000 bases, so 10 times as much DNA that we're looking at, and we looked at that in 1,542 people. So that is roughly a 15,000-fold increase in what we can do in 20 years. Having said that, I would classify this as the type project that we could do at a smaller scale 15 years ago or 20 years ago. We did it. But we can do it at a scale that actually probably matters now. In addition to this type thing, though, there's also things that we couldn't even think of. So this is the study I mentioned with, we worked with Mike on, where we looked at actually thousands of individuals, but looked at all the genes that make up their protein coding region in each individual. And that really wasn't conceivable, in 2005. As another example of that, we're now actually even sequencing routinely whole genomes. We just had a project that had to do 40 whole genomes. One of our collaborators said, well, how are we going to do it? I said, it's already done. We did it. It took like 2 weeks. It's finished. This is a case where we actually took 3 whole genomes from the same patient, normal tissue from the esophagus, Barrett's esophagus, a precancerous lesion, precancerous condition, and esophageal cancer from the same patient, in collaboration with a group at Johns Hopkins, and sequenced the complete genome and identified this gene as a gene that is damaged as the cells progress from normal to premalignant to malignant. And again, this would not have been possible to think of sequencing 3 complete genomes when it cost $100 million each in 2000. Another example, this is a large Scottish family with all the people in red and blue have a major psychiatric illness, and we sequenced the circled ones. There's actually a new generation of people from this family, many of whom also have a psychiatric diagnosis. We're going to do 10 more from the next generation. And basically looking at Mendelian genetics within this family, but at the single nucleotide level. And then lastly, we have begun to really think about how we can apply this technology that I mentioned to these big plant genomes, which are still a real challenge. To wind up the future, this has really changed the way we do biology, and it's still changing. There have been recent announcements about even faster machines that will sequence things even more cheaply. And one of the biggest challenges we face is actually keeping up with the data flow. I showed that graph versus Moore's law. That's all nice and good if what you're saying is, well, gee, the sequencing is getting a lot cheaper. But when you think of the flip side of that that means the computers can't keep up, and that's actually a challenge. So that's one of our big challenges still. And I just want to acknowledge, people here who have contributed substantial funding to these efforts, both government and private. And the private has really stimulated and jump-started these things. And then, of course, Bruce and Jim, through the 20 years I've been here, have been enormously supportive of genomics at the lab, and it wouldn't have happened without that support. And then, of course, our various collaborators here at the lab and elsewhere. And to lead into David's talk, we've been teaching a sequencing course here since 1996. And so those students have had great input, and many of them have come back as visiting faculty members and so forth and collaborators. So we finish there and let David take over. I think